Namaste. Myself Dr. Riva Gupta from JAYOTI VIDYAPEETH Women's University, Jaipur, Associate Professor in Department of Kriyashari, Faculty of Ayurveda Science. आज का हमारा सेशन है सेल्फ फिजियोलॉजी फॉर BMS फर्स्ट ईयर स्टूडेंट्स. The first introduction, the basic organizational structure of the human body is the cell. There are 50 to 100 trillion cells in the human body. Differentiation is when cells specialize. As a result of differentiation, cells vary in size and shape due to their unique function. A typical cell has three major parts: cell membrane, cytoplasm, and nucleus. So first, cell membrane. It is a protective sheet enveloping the cell body, separates ECF and ICF. ECF means extracellular fluid and ICF means intracellular fluid. It is a semi-permeable membrane. Its thickness is about 75 to 111 Armstrong. तो यहाँ पे हम ये स्ट्रक्चर देखते हैं सेल मेम्ब्रेन का. तो दो लेयर्स है इसमें. प्रोटीन लेयर है, एक लिपिड लेयर है. जो प्रोटीन लिपिड लेयर है वो बाई लेयर होता है. टू लेयर्स में लिपिड लेयर, जैसे कि हम डायग्राम में देख सकते हैं कि जो लिपिड लेयर है वो बाई लेयर है और उसमें बीच में प्रोटीन्स भी लगे हुए हैं. साथ में प्रोटीन्स के साथ और लिपिड के साथ कहीं कहीं कार्बोहाइड्रेट्स की चेन भी होती है. Cell membrane electrochemical gradient. Due to selective permeability, difference in concentration of chemicals across cell membrane and difference in distribution of charges across the cell membrane is the membrane potential. जो cell membrane होता है वो selective permeable होता है. Selective permeable means सिर्फ कुछ सब्सटेंस जो होते हैं वही जिनकी नीड होती है, सिर्फ वही सब्सटेंस सेल मेम्ब्रेन के अंदर जा सकते हैं, बाकी उसको क्रॉस नहीं कर सकते. सेलेक्टिविटी होती है वहाँ पे कि कुछ ही सब्सटेंस जाएंगे, जो सब्सटेंस चुने गए हैं सिर्फ वही सब्सटेंस जा सकते हैं, बाकी अंदर नहीं जा सकते. सेल मेम्ब्रेन से न तो अंदर जा सकते हैं न बाहर आ सकते हैं. और इसी कारण से वहाँ पे जो चार्जेस होता है उसका डिफरेंस होता है. सेल मेम्ब्रेन के अंदर और बाहर जो चार्ज डिफरेंस होता है, वोल्टेज डिफरेंस होता है, उसी को हम बोलते हैं इलेक्ट्रोकेमिकल ग्रेडियंट या मेम्ब्रेन पोटेंशियल. जैसे हम डायग्राम में देख सकते हैं कि जो प्लाज्मा मेम्ब्रेन है या सेल मेम्ब्रेन है, उसमें बाहर जो एक्स्ट्रासेलुलर फ्लूइड है वहाँ पे मोस्टली सोडियम आयन एंड क्लोराइड आयन्स है. तो सोडियम आयन ज्यादा होने के कारण प्लस कुछ पोटेशियम आयन भी है, तो वहाँ पे पॉजिटिव चार्ज ज्यादा मिलेगा, तो वहाँ पे पॉजिटिविटी होगा, पॉजिटिव आयन होंगे. एंड जो अंदर है सेल के अंदर वहाँ पे मेनली फॉस्फेट्स आयन और सल्फेट्स होते हैं, प्रोटीन्स होते हैं, क्लोराइड आयन्स है, और जो पॉजिटिव चार्ज आयन है पोटेशियम वो कम है, सोडियम भी कम है. तो इस वजह से अंदर जो होता है चार्ज वो मोस्टली नेगेटिव होता है. तो जो सेल मेम्ब्रेन होता है उसमें भी हम देखें तो बाहर की तरफ पॉजिटिव चार्ज होता है, अंदर की तरफ नेगेटिव चार्ज होता है. देन साइटोप्लाज्म. इट इज अ जेली लाइक मटेरियल. It has two main parts: cytosol. First is cytosol. It's a clear liquid portion, and cell organs. It is made up of two zones. First ectoplasm and second endoplasm. Ectoplasm is peripheral part, and endoplasm is inner part, which is interposed between ectoplasm and nucleus. Organelles with limiting membrane: endoplasmic reticulum, Golgi apparatus, lysosome, peroxisome, centrosome, secretory vesicles, mitochondria, and nucleus. Organelles without limiting membrane: ribosome and cytoskeleton. Functions of cell organelle. First, RER means rough endoplasmic reticulum. Synthesis of proteins. Degradation of worn out means useless organelles. Then SER, smooth endoplasmic reticulum. Synthesis of lipids and steroids. Role in cellular metabolism. Storage and metabolism of calcium. Catabolism and detoxification of toxic substances. Next is Golgi apparatus. It works processing, packaging, labeling, and delivering of proteins and lipids. Then lysosome. Degradation of macromolecules. Degradation of worn out organelles. Removal of excess secretory products and secretory functions. Then peroxisomes, breakdown of excess fatty acids, detoxification of metabolic products, oxygen utilization, acceleration of gluconeogenesis, degradation of purine to uric acid, role in the formation of myelin, role in formation of bile acids. Next is centrosome, movement of chromosome during cell division, mitochondria, production of energy, synthesis of ATP, initiation of apoptosis. Next is ribosome, synthesis of proteins. Next is cytoskeleton, determination of cell shape of the cell, stability of cell shape, and cellular movements. यहाँ डायग्राम देखते हैं इसका सेल का, इसमें न्यूक्लियस शो हो रहा है, न्यूक्लियस में डीएनए होते हैं स्ट्रैंड्स के रूप में, क्रोमेटिन स्ट्रैंड्स के रूप में, देन न्यूक्लियस के चारों न्यूक्लियर मेम्ब्रेन है, and Golgi apparatus, cell membrane, centrioles, endoplasmic reticulum, gel-like fluid है cytoplasm, then ribosomes where proteins are synthesized, nucleus in nucleus, lysosome and mitochondria where respiration takes place and energy is produced. This diagram is showing rough endoplasmic reticulum contains ribosome and lysosome, cytoplasm, nuclear envelope which has pores called nuclear pore, chromatin in form of DNA, nucleolus, centriole, plasma membrane, Golgi complex, vesicles. Next is nucleus. It is largest cellular organelle, diameter 10 to 20 micron. Cells with nucleus is called eukaryotes and cells without nucleus is called prokaryotes. Nucleus is present in all cells of body except red blood cells. Shape is mostly spherical. It is covered by nuclear membrane. Major components of nucleus are nucleoplasm, chromatin, and nucleolus. There is a diagram of nucleus showing nuclear envelope which has pores, nuclear pore, and nucleolus and chromatin, condensed chromatin, and cysteine. And nuclear membrane. It is double layered and porous membrane. Pores are guarded by protein molecules. Exchange of materials between nucleoplasm and cytoplasm occurs through pores. Space between two layers is continuous with lumen of ER, means endoplasmic reticulum. Nucleoplasm, it is highly viscous fluid. It surrounds chromatin and nucleolus enzymes. It contains dense fibrillary network of proteins called nuclear matrix and many substances such as nucleotides and enzymes. Then chromatin, it is thread-like material made up of large nucleus of DNA. Just before cell division, the chromatin condenses to form chromosome. Next is chromosome. Chromosomes are rod-shaped nuclear structure. It is formed from a single DNA molecule coiled around histone molecules. Each DNA contains many genes. Next, nucleolus, a small round granular structure. Each nucleus contains only one or more nuclei. Nucleolus contains RNA and some proteins. Functions of nucleus: control of all cell activities that include metabolism, protein synthesis, growth, and reproduction. Synthesis of RNA, formation of subunits of ribosome, control of cell division through genes, storage of hereditary information and transformation of this information from one generation to next generation. Next is cell death. It occurs by two distinct processes. First is necrosis and second is apoptosis. Necrosis or cell murder. It is uncontrolled and unprogrammed cell death due to unexpected and accidental damages. Apoptosis or cell suicide. It is natural and programmed death of the cell under genetic control. Next is cell adaptation. Changes taking place in a cell in response to environmental changes, cellular adaptation occurs by any of the following mechanisms. First is atrophy, means decrease in size of cell, and hypertrophy, increase in size of cell, aplasia, decrease in number of cells, hyperplasia, increase in number of cells due to increases cell division, dysplasia, abnormal change in size, shape, and organization of the cell, and metaplasia, replacement of one type of cell to another type of cell. Next is cell junctions. Connection between the neighboring cells or the contact between the cell and extracellular matrix is called cell junctions. It is also called cell membrane junction. It is of three types. First, occluding junctions, communicating junctions, and anchoring junctions. First is occluding junctions, which prevent intercellular exchange of substances like tight junctions. Second is communicating junctions, which permit the intercellular exchange of substances like gap junctions and chemical synapse. Next, anchoring junctions, which provide strength to the cells by acting like mechanical attachment, like adherence junction, focal adhesion, desmosome, connection between cell to cell, and hemidesmosome, connection between cell to matrix. Next, tight junction. It is also called zona occludens. It is the region where the cell membranes of the adjacent cells fuse together firmly. Next is gap junction. It is also called nexus. It is the intercellular junction that allows passage of ions and smaller molecules between the cells. Next is chemical synapse. It is the junction between a nerve fiber and a muscle fiber or between two nerve fibers, through which the signals are transmitted by the release of chemical transmitter. Next is adherence junction. It is the cell-to-cell junction which connects the actin filaments of one cell to those of another cell. Adherence junction provides strong mechanical attachments of the adjacent cells. Focal adhesion. It is the cell-to-matrix junction which connects the actin filament of the cell to the extracellular matrix. Next is desmosome. It is a cell-to-cell junction where the intermediate filaments connect the two adjacent cells. Next is hemidesmosome. It is a cell-to-matrix junction which connects the intermediate filaments of the cell to the extracellular matrix. So there is a diagram of junctions showing tight junction, hemidesmosome, desmosome, focal adhesions, gap junction, adhesion belt. Here is some questions for students. Describe cell, define nucleus and its functions, define different cell functions, mention cell organelles and their function, and prepare model or project of cell mentioning its organelles. There are some reference books. Thank you.